Hello, this is Martha Nance, and we're back for the last session of today's Family Day presentation, which is your opportunity to ask questions of these elite group of researchers. You just heard them give descriptions of some of the research studies that are ongoing or are planned to start soon. And here's your chance to ask people questions. The first question, I think, is kind of a complicated one, and I think I'm going to start with Ed Wild on this. So, in one of our scientific presentations yesterday, we heard that it might not be possible to precisely measure the amount of mutant Huntington in the spinal fluid because of the expansion of CAG repeats in the brain cells, and if there's a range of repeat numbers, then that somehow messes up the CSF measurements. How is that? What have you found, Ed, in the HD clarity in the spinal fluid studies?
Yeah, it's a question for Tacey, and it's a really good one, a really important one. And I think it really emphasizes the fact that all of the Huntington assay work is a work in progress. The first assay was described in 2015 that was sensitive enough to measure mutant Huntington in spinal fluid, and that's been a really important measure for the Huntington lowering trials. But as we heard yesterday, there are technical issues to do with the way the number of CAG repeats can sometimes change in the brain during life that could make it much more complicated. I think the bottom line for this particular question is that it's probably not a deal breaker for the purpose of measuring whether we're lowering Huntington in the clinical trials. And that's because the key thing is how much each individual person's Huntington changes from the start of the trial to the end. In other words, did it go up by 10% or down by 10%? And over the course of a trial that's 1, 2, 3, even 4 years, it's very unlikely that that individual person's CAG repeat length would change. And so the measurement comparing the beginning of the trial to the end is likely to be meaningful. It may mean that we can't necessarily say you had 62.5 femtomoles at the beginning and 61 at the end. But it certainly overall gives us a really reliable and meaningful measure, I think. There's a lot of debate about exactly what it means and what it means if it goes up or down. But I think broadly speaking, for a Huntington lowering treatment, down is good and up is bad. I think the other thing to say is that one of the biggest things that HD Clarity is aiming to do is to provide us with the big volumes of CSF that we need to produce better assays and to produce a whole suite of measurements, not just for Huntington, but for other things that will help us figure out what's going on in all of these trials. And CHDI and others are working on making things like total Huntington or all of the little chopped up bits of Huntington, making tests for those that can be deployed across the trials so that if everything is moving in the right direction, it doesn't necessarily make such a big difference if one of the assays has a particular shortcoming in one particular area. So strength in numbers is, I think, the message across biomarkers.
Okay. And Dr. Levitt or Dr. Eileman, any comments from the perspective of studies that have been happened already or are just about to happen, how you're going to deal with this issue of the change in repeat numbers?
I think Ed's answer was very clear, and certainly during the time course of the trials that have been ongoing, we don't believe this is likely to be a significant confounder, or it's not going to complicate the results within an individual when we're comparing, pre-dosing and after treatment levels. So I don't think it's a significant concern the way that the assays are being used.
I would fully agree. I mean, the early stage studies that we're doing currently in the WAVE program obviously take about 4 to 6 months, and even the Generation HD1 study was aiming to do 2 years. So I think in those periods, and with then those higher numbers of participants, I would also expect that we could use those assays.
Okay. And Dr. Eileman, since I have you online right now, is there any progress? We know that the presentations everybody gave were pre-recorded. and you were thrilled that you had just dosed the first person. Have you dosed a second person or a third person? Is there any more progress on that study, the Wave 3?
Yeah. I was just laughing myself because it's obviously Saturday here today. My team wouldn't be very excited to dosing the first patient today. This was the first patient in Münster, by the way, as I mentioned. So the first patient in the study globally had been dosed in September. And since the study was just started, I think we have a good dynamic. Obviously, as I mentioned earlier, for this particular study, you need this specific genetic marker, the SNP3. So what is going on largely now in the participating sites, some of them have just been activated now or are in the process of being activated, is to pre-screen patients to see whether they have that specific SNP3 characteristic. What is good news, I think, is that those participants that previously did participate in the Precision 1 and 2 trials, the pre-screening now is much faster. In the other two studies, we have experienced that it sometimes took quite long to get the results. So this has been much improved. So there was good progress now. The phenotyping really goes very quickly, and we have the results within 2 to 3 weeks, as it looks like. So that's very encouraging. It's good progress, and we expect the study to enroll actually quite fast because we have multiple sites, more sites than in previous studies participating. And in spite of the fact that obviously the SNP3 only is available in part of the population, we are hopeful that the study can progress quickly, and with this new chemistry that we might have new exciting results. Hopefully we'll have to see. I think it's an interesting experiment because of the allele specificity, at least the targeted allele specificity. So we'll have to prove that it's actually allele specific, but I think the chances are very high looking at the preclinical data, and that should be interesting to see. So it will probably be a quite dynamic progress, and we'll report soon on that.
Okay. There are several questions for Dr. Levitt about branaplam. Number one, how is it different from another company that's developing a similar kind of product, PTC 518? A second simple question is, will the branaplam study exclude people who participated in Generation HD1 or the Wave 1 and 2 trials? And then the third question is, how do you find out what sites are going to be involved in branaplam? So 3 questions for Dr. Levitt.
Sure. And I'll start with the first question. So both branaplam and the PTC drug, PTC 518, are small molecules. They're both orally bioavailable. In other words, they're both medicines that are taken by mouth. They get into the circulation, and they both are brain penetrant small molecules, so they get to the brain. Based on my understanding, both of them do target splicing, and they actually both target in the Huntington mRNA splicing at the same exon sites. They are different compounds, so they all have slightly different what we call pharmacokinetics and pharmacodynamics. In other words, they'll be absorbed at slightly different rates when they're delivered orally. They'll have different levels in the blood, and that will lead to potentially different levels in the brain and the spinal fluid. So they are different compounds. They will have different properties. This will likely lead to different levels of dosing, and it may lead to different levels of efficacy. And ultimately, this may influence the different side effect profiles. So they have similarities and they have differences, and both should be studied in appropriate double-blind, well-controlled trials. The second question, I did quickly review the current exclusion criteria in the protocol that I have, and it does define that individuals who previously participated in Huntington lowering experimental therapies, which would include Generation HD1, any of the Roche studies or Wave ASO trials, those individuals will be excluded from Vibrant HD. Now the study has not actually started, and this may be something that changes. So ultimately, at the end of the day, the final protocol will determine. But at the current time, my understanding is individuals will be excluded. And the best way to find information about clinical trials is to communicate with your local center of excellence, whether it's an HSG or EHDN center of excellence. But also clinicaltrials.gov will have a list listing of that, and likely the HSG will generally have listings of sites for different trials. But we don't have the final list because the study hasn't actually started. So those sites will be available in the future.
And the different sites are still beating each other up to decide who gets to be the one participating in this trial. So,
may I make a short comment, Martha? May I make a short comment there? Because we were just initiated for the study, and actually it was a discussion point that came up here. I do think the sponsor is contemplating, at least on clarifying, maybe in a future protocol version or in an administrative letter, that people that were on placebo in the previous ASO trials possibly will be able to participate in the study, to the best of my knowledge. So there may be a window of opportunity here for those people that after the Generation HD1 trial has finished and will then get their information on their status. If they've been on placebo, there is no scientific rationale for excluding them. So there is a potential for those subjects to participate, which is already predefined in some of the other protocols. And I think it's going to be for Vibrant as well. At least that's the intention, to the best of my knowledge.
Okay. 2 short questions for Dr. Wild, who I know has to catch a train or cab or something soon. Are you looking for volunteers for HD Clarity who are not at risk for HD, or is it only for the people who are known to be gene positive or known to be affected? And then the second more important question is, what is your dog's name?
The dog is called Riley. If you can call him a dog, he's the smallest possible dog. I'm cuddling him because there's fireworks going off outside for Guy Fawkes tonight, and he's terrified and has a very tiny brain. But he's enjoying the cuddle. The answer to the other question is yes. Emphatically, we are very much welcoming healthy controls. So that would be people who are a friend, partner, spouse, acquaintance of an HD family member, or someone who's had a negative test for the HD mutation. So the healthy control arm is, as ever, is really important for figuring out whether the changes that we see in the people who have the mutation are disease related or coincidental sort of changes, or age related, or that kind of thing. So yes, very much so. Please inquire at your local enroll site.
And Dr. Furst-Deming or somewhat one of your group, has Unicure started to enroll further patients into the Highdose Cohort? And if not, when was that going to happen?
I can answer that question. And thanks for the interest. And we're currently in the Highdose. We are enrolling a set of 12 patients right now, and we are eager to hear from you. So contact your local site and ask about information and how you can help. So.
Good. And another really important question for anyone who wants to answer. Most of the trials for HD are for just diagnosed or even people who haven't been diagnosed yet. Is there anything coming for people in the mid or later stages of Huntington's disease?
Well, maybe I'll take that one. I do think, many of the trials that we currently do have exclusion criteria that limit participation to subjects to TFC stage 7. Which basically includes stage 1, stage 2 Huntington's disease. That's appreciated. Mind you, for a lot of these trials, we do need patients to be able to come to the centers, which kind of defines these levels. But I do understand that a lot of discussion is going towards prodromal Huntington's disease and to even earlier stages. That's appreciated. I do think, and please know that I think all the investigators, all the clinicians are mindful of that, and we are talking to sponsors that while you might have to do validation of a proof of concept of certain therapies in early, maybe even prodromal stage of Huntington's disease, if we are successful with these trials in the patients that need to be flexible, go to MRIs and stuff like that, I'm very certain that there is high interest of everyone to try these compounds also and these treatment approaches in different stages of the disease. That also, by the way, includes, and I think that comes very much to my heart, and I know Martha is involved in that area very much as well. And please be certain that all of us also are very much thinking about the juvenile Huntington patients, which unfortunately in most of these patients, most of these protocols, most of these studies are also excluded for certain reasons of doing these studies very rigorously. We have that in mind, and we are trying to look into the feasibility of also conducting ASO and gene therapy trials in the juvenile population. So there have already been attempts to write protocols and to think about how to organize that. There is a specific working group which is jointly conducted between the European Huntington's Disease Network and the Huntington Study Group. And many of us are part of this working group. And I think a lot of thought is put into these options, and we will not forget it. But I do appreciate that currently for the more advanced stages, there is a very small, usually only very local trials, if at all, offer for participating in trials.
Okay, thanks. And
Martha,
just a comment.
If I could just
go ahead.
Sorry, if I could just have a follow-up. I have had confirmation that what Ralph had suggested is the case, that if an individual was participating in a Huntington lowering trial previously and they were on placebo, they will be allowed to enroll within the Vibrant HD study. So just a point of clarification there that now has been included in the protocol moving forward.
And again, that will be true also for I think pretty much all the other studies, because it makes sense. But nevertheless, by the way, I think we all very much, just to say that we would like to ask you to remain in the Generation HD1 study if you still are. It's only a few more weeks to go for most of you, of those that haven't finished the study yet. I think it's very important to learn as much as we can from that huge project. And I'm sure and certain that we will learn a lot of very, very important messages from that study. And the more data we have, the better it is. So I think for the whole community, it would be great if those people that are still in the trial completed. And I'm certain you've heard about the progress of the other programs that, for instance, the WAVE, the Novartis study, there's going to be a PTC study, as we all know, coming up. There will be multiple trials, the Unicode trial, of course, that will still be recruiting. So there will still be options for you if you were on placebo.
And a quick question for Mikhail, who's standing in for Michael Hayden. Can a person enroll in the open label extension in the Proof HD trial, the Prodopidine trial, if they have not already participated in the double-blind portion of the study?
Yes, thank you for the question. Just to send regards from Michael. Unfortunately, he was unable to join as he's traveling. Currently, only participants that are enrolled into the Proof HD trial are able to then enroll into the open label study. We do understand that there are many patients that were not eligible to participate in Proof and do want to take this drug. We are looking into some options and considerations, but currently only participants of Proof will be eligible into the OLE.
By the way, I would like to interfere here. Apologies, but I do believe, actually, being one of the PIs of the Proof study, that Prodopidine, for instance, would certainly be a compound that, if it was successful in the Proof HD study, as we all hope, that would be a compound that I would think could be fairly easily also trialed in a more advanced stage of Huntington's disease. It's rather easy to apply. It's an oral preparation, and we have actually trialed it in more advanced patients in the past. So the earlier studies, HEART and MIRM-HD, allowed more advanced patients into the trials as well. So let's see what happens, but I do think that this would potentially, in the case of success of Proof HD, be a treatment approach that we could move to more advanced stages rather easily.
So do any of these studies, any of you have any plans, including HD Clarity, I guess, to include juvenile HD? It looks like every study I'm reading says 20 and above, 25 and above. Ed's got a thumbs up. Go ahead, Dr.
Yes,
Wild.
we just submitted an amendment in the UK, which we're hoping to roll out globally, to allow us to begin collecting CSF in HD Clarity from patients with juvenile HD down to the age of 11. Now, clearly, this is not something that is going to be, I mean, juvenile HD is very rare, and within that population, it's very rare to for an affected child to have the enthusiasm and understanding and altruism, I guess, to be able to take part in a study of that kind. But hopefully this, and I think Pagnopoulos has really blazed a trail where essentially parents know their children best and know what they want to do. And in many cases, kids would want to help other people in a similar situation. I think that's something that the research framework needs to allow. And that is what we hope to open the door to in terms of spinal fluid collection. And with that, my taxi driver's calling and I have to go. I'm so sorry. I love you all very much. Bye-bye.
Bye, Ed.
Dr. Wild.
I would like to add to that, actually, if I may briefly. I just want to clarify that this is also not something that sponsors can just do or not do. At least in the European Union, there is a regulation that if you want to advance a drug or a treatment to approval, you actually have to have a pediatric development plan. There are some exempts to that. So in the past, actually, Huntington's disease was excluded from that, but that has changed a few years ago. So there actually is a need for any sponsor that wants to get approval for a drug in the European Union, at least. So I don't know exactly what the situation is in the United States at the moment. Apologies for that. But in the European Union, the sponsor has to provide a pediatric development plan. So that is very carefully checked. And for that reason, also, we have the EHDN and HSG Juvenile Working Group that helps sponsors to address that question.
Okay. I have a question for Dr. Koenig, which is, you said something in your presentation that you're really excited about getting patient and family input into your study. And what specifically have you done or are you doing to get patient and family input? And by the way, do any of the other studies include patients or families in the planning process?
Sure. Can you hear me okay? Great. Yeah, I mean, I think a big focus of our program is on functional outcomes, and that can be collected in a number of different ways. And so we encourage having study partners as part of our trial, and that will include scales that they complete as well as patients collected. In terms of the planning of the trials themselves, we are very involved in seeking the feedback of both those affected by HD as well as families. So I've tried to structure the assessments that way. So and a lot of it is focused on cognition and things that patients may not have immediate insight into, but there may be changes that are more subtle that others might pick up on. So thank you for that question.
Yeah. And I'll just ask a couple of questions for Aaron and Sarah from Unicure. First of all, did you include patients or families in your planning process as you designed your study? Number one. Number two, we hear that you're including European sites. How about more North American sites?
This is a wonderful question. Okay, well, I'll have to say that Aaron is one of our wonderful sites, and Ralph is another one of our wonderful sites that will hopefully be opening up soon in Europe. But we have opened up our first European site, and so we're very, very happy to announce we just screened our first patient. So thanks to everybody who worked so hard to make all of this happen. And that site is in Poland. And so we're very, very excited to be working with the group in Warsaw, and it may even be that some of the patients who are screened, some of the first surgeries will be done in Poland. And so stay tuned, and so we're excited to get that up and going. In the United States, we have just opened up a couple of other sites, so stay tuned and look at watch clinicaltrials.gov for more sites that will be opening up. And the other thing that is happening is we are being contacted by families, and certainly it makes a lot of sense to And one thing that we do ask from our patients and families is contact your patient and family and spread the word, not just about our trial, but all trials, because we all need to work together in order to understand better what's happening with our trials and our treatments, so that we know what to do. So trial Europe is our exciting announcement, and more sites in the US as well. So thank you.
And then one of the unusual things about your study is that there's a site maybe where you are screened and followed up on, but the surgeries are not done at every site. There's only 1 or 2 surgical sites. Is that correct?
Exactly. And I think that's one of the great strengths of our study, in that what we're doing is, as we're all learning together about sort of the optimal way to perform these surgeries, we can do it in a very, very controlled environment by making sure that our surgeons are well trained and they all work together. What we have is we have a neurosurgical planning committee, and all of the surgeons in the trial. We currently have 3 surgical sites in the U.S., and we currently have the 1 surgical site in Europe, and we hope to be opening more. But all of the surgeons are working together so that they see what's been happening to all the surgeries. So even the European surgeons have been following the American surgeries, and so we are all learning from each other. And then so by having that tight-knit group who are sharing their expertise and sharing their experience, we're hoping to perfect this technique.
Actually, if I may briefly interrupt here as well, I think this will be very interesting, and I think the whole study team is committed to also publishing in the future as soon as possible the learnings from the setup of this trial. We won't go on the details here, but you know about like the screenings in the beginning and everything now, and also the volumes to be considered. There's a lot we learn while we do this trial. And of course, we appreciate the input of so many people that have helped us successfully going where we are. And I just want to briefly say thank you, guys, that you understand that we want to have a few centers in Europe as well. The patients here want to participate in that approach as well. And we are very happy that Unicure gives us the opportunity to start the trial here as well.
And there was a question about whether PTC has started its trial and how can you sign up. And I would, again, encourage for trials that we're not discussing in this particular research blitz, go to clinicaltrials.gov, go to hdtrialfinder.org, and find out about those trials as they get started.
But I think I can declare that we already had a pre-study visit, so things are moving also for PTC.
Yeah, so it should be up in clinicaltrials.gov, and so there should be sites that you can identify. I had one audience member say thank you to Sage for taking on cognition. Nobody's ever done that before in Huntington's disease, so bless you. Thank you.
There were trials.
Yeah. And then another question, I'm going to go for, maybe 4 more minutes, but I'd like this erudite group of people to speak about something we learned in our scientific meetings about biotin and thymine. What do you guys think about that? There was a discussion about these 2 vitamins as being particularly important for function in this part of the brain, and that maybe treatments would be helpful certainly in animal models of HD. What do you guys think of that? Stunned silence. How about Aaron first inning? No, Let's let Aaron speak for a minute.
Well, yeah, thank you. I hope that everyone had the opportunity to watch Sarah Pico yesterday. If not, I would urge you to watch her on demand, her really eloquent presentation about the work that they've done in evaluating thymine levels and biotin levels in mice, but also in humans in the CSF, cerebrospinal fluid. I believe they are planning an open label trial actually to evaluate the efficacy of high-dose thymine and biotin, but it hasn't been done yet. So the short answer is, we don't know. More to come. We don't have enough evidence to recommend biotin or thymine at this point, but hopefully we will have an answer once they complete their trial.
And no one has any dissenting views. Okay, we have a couple other questions. Let's see, nobody's here from Connect. Well, maybe Aaron knows about Connect also. In the valbenazine trial, are we specifically measuring changes in swallowing?
Oh, that's a really good question. So we are certainly including. We want to ensure that individuals are okay with swallowing and not struggling with swallowing prior to entering the trial. And so that's not a primary outcome or secondary outcome measure, but certainly something that we are watching and will hopefully be able to comment upon once we complete the analysis of the trial. It's a really good question and an important question, because I think there's controversy and we don't really know what the impact the VMAT2 inhibitors have on swallowing.
And then one final question maybe is a broader question about how we interact with regulatory authorities. We were asked, is Novartis going to file for, quote, orphan designation in Europe? And what in the heck does that mean anyway? And does that somehow get the study done quicker? Is that true in the US as well? Anybody want to comment about how we interact with regulatory authorities as we set up our studies?
I can address the specific question. So Novartis has filed for orphan designation in the US and in the process of applying for it in Europe. And in general, what this does when it's approved is does shorten some of the regulatory timelines and accelerates some of the interactions with the regulatory agencies. So long, complicated answer to yes, it does shorten the time it takes from completion of studies to approval of drugs. So it is a huge advantage to the field that we can obtain this orphan status, and yeah, I think it's appropriate, and certainly Novartis has pursued that for the Vibrant HD study and for branaplam.
And by the way, this is generally for rare diseases, and it also has a specific, at least in Europe, a specific development plan for pediatric populations, which means if you go to pediatric development, you may even need less evidence, less sample size and trials, maybe only one small trial and things like that. So that will actually help us to address one of the issues that we discussed earlier.
And is this orphan designation true also for Unicure or Sage or Prolenia for your guys'
It means
studies?
rare disease. It means a rare disease, and Huntington's disease is per definition, with the incidences even that we know now, a rare disease. So orphan designation applies to Huntington's disease generally. Yes.
Just in general. Okay. I think we'll wind up. We're 12 minutes over time, 13 minutes over time. And thank you so much to the researchers. Thank you so much to the patients and families who came to this Family Day. I hope it was a wonderful experience for all of you. I certainly learned a lot of things. And next year, we're hoping to be in person again. But this is certainly better than nothing. So round of applause and thanks for all the participants today, and have a great rest of the day and a good rest of 2021. Stay healthy for 2022.
And thank you for supporting many trials we are doing so well. Thank you, guys.
Yep.
Bye-bye.
Yep.
Take care.
Thank you, everyone. Bye.
Take care, everyone. Be well.
Love your glasses.